Good morning to all of you. Today we are going to perform the experiment number 1. The aim of this experiment is to study the microscopical, macro and microscopical characteristics of clove. So what do you mean by macroscopical? As the term denotes, macro. Macro means you have to observe the gross structures or characteristics of the clove. Right? You have to observe the color, order, shape and whatever you can observe in the morphology. And what we are going to do in the microscopical studies, we are going to do the TS transfer section studies. So first of all, we will study the macroscopical characteristics of clove. So what is clove? Clove is a dried flower bud obtained from the plant Eugenia caryophyllus belonging to the family Myrtaceae. So why we should study the clove? Because clove is medicinally important and it is a good dental analgesic. Due to presence of clove oil, the clove oil at least it should contain 15% of clove oil. So first of all, we will study the macroscopical studies. In this, what we are going to observe? We are going to observe the morphology of the flower bud. So this is a flower bud. As you know that flower contains 4 valves. What are the 4 valves? Calyx, corolla, androecium and gynoecium. So all the 4 valves are present here also. You can see this dome-shaped structure. This dome-shaped structure is corolla, and these are the petals. Petals coming under the calyx. So corolla, petals, and these are the sepals. And this stalk is called as hypanthium. The scientific name is hypanthium, which contains the oil glands. So clove oil is present in the hypanthium portion. And this is the ovary, gynoecium. In the gynoecium ovary is present, and ovary is present just beneath this. In here, if we can see, the ovary is present in this region. Whereas in the LS, this is the LS is the longitudinal section of the clove. So ovary is present here, and in ovary ovules are present. So when we will observe the LS of the clove, what we will see? We will observe the style. What is the style? The style is the portion of gynoecium. So gynoecium contains ovary, style and stigma. If you are remembering, it is like this. So this is the style. The stalk of ovary is called as style. So this is the gynoecium. Gynoecium contains ovary, style and stigma, and the style is this. And what are these? Pollen grains are present here. What are pollen grains? Pollen grains are the part of androecium. So all the 4 valves are present here also. Stamens, style, petals, corolla. These are the sepals. So sepals are the part of calyx. First one is sepal only, and oil glands are present here also. And after the ovary, there is a structure present called as columella. So what is the columella? Columella is nothing. It is a support for the ovary, or you can see like that ovary is sitting at the columella. So when we will study the microscopical studies, we have to study or we have to observe the 2 sections. First section, the section through the ovary, and another will be section through the columella. So we will, by the study of transverse section, we can identify which section is contain the columella and which section contains the ovary. So when we will study the microscopy of the clove, you are going to observe the 2 sections. One section will be the section through the ovary, and another will be the section through the hypanthium. So we will study what you are going to observe in the section. Outermost there is the epidermis, followed by the oil glands, and this space is called as cortex region, whereas 3 concentric layers are present above the ovary, and these concentric layers are of vascular bundles. So 3 concentric layers of vascular bundles are present above the ovary, and in the center portion, in pierced through the ovary, in the center portion you are going to observe the ovary, and ovary contains the ovules which further convert into the fruit or the seed. The ovary will convert into the fruit and ovules will be converted into the seeds. So in the section through the ovary you are going to observe the epidermis, oil glands, oil in the oil glands, oil is present. Clove contain at least 15% of oil, clove oil, and after that this region is not the blank. This region is called as cortex. Cells are present, but we have not shown because normal usual cells are present, and special cells are three concentric layers of vascular bundle above the ovary, and in the ovary ovules are present, and this center portion is not the blank. Cells are present, tissues are present here also. When we will study the section through the hypantheum, there is a little difference between the section through the ovary and the hypantheum. In the hypantheum region section, you are going to observe the blank central region. You are not going to observe the ovary here. So you will identify this section is section through the ovary and that section is through the hypantheum. So in this region, you are going to observe the blank or the arendthymal tissue. So in the arendthymal tissue, central portion columella is present, and arendthymal, how you are going to identify the arendthymal cells? The cells will be at the distance and light will be coming from that. That I will show you in the TS. Columella is the central region, followed by the arendthyma, and only one concentric vascular bundle ring is present. Whereas in this section, three concentric vascular bundle rings are present. So this is the difference. You can easily identify this section is through the hypantheum and this section is through the ovary. And followed by the cortex region, same, and eye glands, and in the last, epidermis. So there is a similarity and a difference. What is the similarity? Similarity is this: outermost region is called as epidermis, followed by the oil gland cells. So here also our oil gland cells are present, and cortex region. In the center portion there is a difference. In the columnar section, in the hypantheum section, you are going to observe the one concentric ring of vascular bundle followed by arendthyma and the central columnar region. Whereas in this section through the ovary, you are going to observe the three concentric layers of vascular bundles, ovary and ovules. So these are the two sections you are going to observe and study, and you have observed columnar in the LS also. So what we have studied till now? We have studied macroscopical, microscopical characteristics of clove, but on the board. Now we are going to study going to perform the practical, and we will observe in the microscope. Between one index finger and the thumb, and now we will cut the tears. You are not going to do like this, you have to do like this only. See how many sections are coming. So thin section you have to select. We have to cut the sections till here because ovaries will be here only. These are the sections containing columella or the hypantheum. The blade should be sharp, otherwise section will not come. So you have to cut till here. Now these sections came. You can see in the watch glass, few sections are thin or thick. So you have to select the thin sections. So for what? you have to remove all the thick sections. Take a slide that should be clean and dry and select thin sections like this. You can keep 2 or more sections in one slide at a distance. The sections should be full section. That should not be pieces like this. These sections these are the thin sections, but you are not going to select. We are not going to select these sections because we will not observe the whole structure. Okay, we will select the complete section like this. So we have selected one section and that we are going to stain. So how we are going to stain? Usually we will take the saffronin as a stain. And because this is a concentrated solution, so we have to dilute it with the water. Now we will keep this section here for 2 minutes and then thrice we have to wash it with the water. We have to transfer it into water. This is a water and we have to discard the solution. First wash glass, second wash glass with the water and third with the water. So that it should not take more stain, it should only color. First, what we are going to do, how we are going to prepare the slide, we will keep this section which we have washed thrice with the water, we will keep like this and we will put 1 drop of glycerine here. You have to make the 45 angle first. Exactly you need needle, but now you can do like that also. Slowly we have to keep. See, first you are going to like this. Now slowly you are going to keep the cover slip. Why are you doing like this? Because if you are keeping directly cover slip, air will entrap and air bubbles will come. Now we are going to observe in the microscope. So we have observed the tears of clove through the ovary as well as through the hymenthia in the slide, and all the sections or the parts has been seen as described before. So in this way we can perform the experiment. In the next experiment, what we are going to do? We are going to do the powder study of clove. So what is the difference between the powder study and tears? In the tears you can see each and every cell as well as section, whereas in powder study you cannot see the each and every tissue or well arrangement. So in powder study what you are going to see? You are going to see the oil grains. As it is visualized in the tears, the oil grains were pink in color. Why it is pink? Due to the presence of oil. And in this region, cells were present, which is called as cortex. Okay.